Welcome. I'm Erin Stretz. I'm the Assistant Director of Science and Stewardship at the Watershed Institute. Our StreamWatch Program Coordinator is Gian Smith, so you will have heard from her to get your site assignments and some other kinds of communication, or perhaps from Eve Niedergang, who's our volunteer coordinator. Gian is out on vacation, so I am stepping in for her, but she will be back tomorrow to respond to some of your email questions and to, of course, do the lab work for all of those samples that you'll be bringing in tomorrow. So our agenda for today is to go over the visual assessment that you will be performing in the field using Survey 123, which is an online form that you'll use to run through a series of questions and submit that to us digitally. Then we'll talk about harmful algal blooms specifically, because that's a big part of the visual assessment itself. And so I want to make sure that you guys are comfortable with what a harmful algal bloom looks like so that you're able to report it. We'll talk about the water samples that you'll be collecting for E. coli, nitrate, phosphate, turbidity, and chloride. How you'll collect them, how you'll transport them to the watershed center, and then how you'll deliver them. And then we'll talk about kind of what happens after that. So just first, a little bit about the Watershed Institute itself. If you're not familiar with the Watershed Institute, our organization has been around since 1949. We started as the Stony Brook Millstone Watershed Association. And I believe we changed our name in 2018 to better reflect our ever expanding goals. Our targets were kind of expanding from a regional focus more to a statewide focus. So we renamed as the Watershed Institute. But our mission is pretty much the same, and that is to keep water clean, safe, and healthy for ecological health and for the humans who are interacting and playing in that water. And we tend to do this in 4 different ways. The first way is through conservation. So here, I don't know if you've been to our property, we've got about 1,000 acres of preserved land with over 10 miles of trails. And so we invite you to come out and check that out. But, you know, beyond being kind of a recreational hub, the Watershed Reserve being protected land, it's prevented from additional development occurring on the property. That therefore rainwater, when it falls, it's allowed to percolate into the ground as it was supposed to naturally. So we are kind of providing some of those ecosystem services to help treat some of our water before it, gets back into the water cycle, either through groundwater or through our surface waters in rivers and lakes. We also, of course, do a lot of environmental education on our property and abroad. So we have school groups who come to the Watershed Institute. We have educators that go to schools. We also do programs for adults, for preschool, for everything in between. Right now it is summer camp season, so we've got hundreds of screaming and laughing children here every day during the week, which is also just lovely. Hear the screams of people and tie-dye, it's fun. So we have a team of advocates as well. And I like to think of advocacy as just kind of enhanced education. So we're taking what we know about the environment and what we hope to see as far as like improvements in environmental protection, and we take that statewide, we take that to municipalities, and we advocate for stronger regulations so that we can continue to enjoy this beautiful place here on our reserve and also throughout the state of New Jersey. And lastly, we rely heavily on science, scientific research and monitoring to be able to really give the meat to our advocates so that they kind of know what's going on so that they can act accordingly. We also inform our educators about what's going on so they can teach this science to kids so that they can kind of be the next generation of STEM. And we also, of course, implement science in our conservation work as well to track invasive species and native species and deer and all of those things. So you are now joining our science team. Our science team consists of, I think, 5 staff members right now. So there's no way that we could possibly get out to all of the monitoring sites that you will be able to get to every week. So you're really helping us out to fill out this picture of health throughout New Jersey. So our StreamWatch team, that's our probably most visible element of our science program, and that's what we're talking about today. Our StreamWatch team is broken out into 3 different teams. First, we have the chemical action team that was started back in 1992. And with that, we have a team of volunteers who go out to their sites every month. They do a series of chemical tests and they submit that data back to us. And that's kind of independent work. They have almost looks like a briefcase that has all of their test kits in it. And they go out and submit their data, and they actually come into the Watershed Center just about once a year to go through a series of tests to kind of show that they know what they're doing when they're out in the field. So that just provides a little extra layer of quality assurance for us. Our biological action team, they head out into the field twice a year in the spring and fall, and their goal is a little bit different. They use nets and to flip over rocks, to kick the bottom of the stream, kind of zhuzh up that substrate. They will use the nets to scrape along leaves and twigs and logs and things. And their goal is to find macroinvertebrates, which are little organisms that live in the stream. They could be insect larvae, they could be crustaceans, they could be mollusks, worms, any one of these organisms that are large enough to be seen with the human eye and do not have a backbone would fit into that category of macroinvertebrate. And macroinvertebrates can tell us a lot about the general health of a water body. Each taxa has its own pollution tolerance value. So that's from 0 to 10. We can basically score our population, our community of macroinvertebrates. We can score it and say, okay, we have a pollution tolerance value of, let's say 4, And so 0 pollution tolerance value means that that organism is going to die off if there's an influx of, sedimentation or some sort of extra nutrient getting into that stream. We're not going to find those organisms. On the other end of that scale are organisms with a pollution tolerance value of 10, and they can live anywhere. And so ideally, we want to see a diversity across that whole spectrum of all different kinds of macroinvertebrates. But we do definitely want to see those taxa in the lower pollution tolerance value, because that we can infer from that the stream is doing pretty good. But with this group today, what you are joining is the bacterial action team. And so for you'll be going out to monitor and collect samples for weekly, every Sunday for a 5-week period. So we have had this summer, we've had 3 5-week periods that volunteers have been a part of. And we are just starting into session C, which is our 3rd period and final period. So that's where you'll be joining us. So every Sunday, you'll head out to monitor, collect samples, bring them back to the Watershed Center for us to analyze. And you're also keeping an eye out for HABs, or harmful algal blooms, and we'll definitely talk about that. So our sites are all across kind of the western part of central New Jersey. This covers the Millstone watershed, the Stony Brook watershed, and this kind of middle range of the Delaware River watershed as well. So we kind of have these 3 different sub-watersheds that we are working in. And it goes all the way up to Manville, where the Millstone River flows into the Raritan River, all the way up to Lambertville on the Delaware River basin side. And then to the south, we go to Crosswicks Creek. And then to the east, we're in Millstone Township as well. So we're kind of hitting all of these 4 corners of our coverage area. I think this group of people, we've got a few people who weren't able to make it today, but this is our largest group that we've had so far. And we're able to get to 48 monitoring sites every week for this session C. So I'm really excited to see what comes out of that. And of course, if you're looking at our maps on our website, we have color coding to indicate, the general quality, the condition of each of these sites. Green means good, red means bad, and so we've got lots of these maps, and we'll definitely share this with you so you can be able to track your data as it comes in. And so you can kind of see where it fits into the bigger picture as well. So our session schedule, again, we're in session C. We're at training right now. It's July 30th, 10 a.m. Our sampling session will start tomorrow, July 30th or 31st, and go for 5 Sundays. The last Sunday will be August 28th. So the timing for those dates, we would love for you to get your samples to the Watershed Center dropped off by noon at the latest. So that means that you may have 2 sites that you're hitting, you may have 3 sites. You want to kind of time it so that you head out in the morning, you travel around to your 2 or 3 sites, and you have timed it so that you're here at the Watershed Center by noon. So this first session tomorrow, of course, this will kind of be a learning experience. This may be the first time that you're visiting these particular stream sites or lake sites. So it may take you a little bit longer to kind of figure out your route tomorrow, but going forward, I anticipate that it'll be a little bit more streamlined, and you'll kind of get into a groove and figure out your own schedule. If you are ever can't make this time, or you can and you just think I'm going to be running a little bit late, I think we can accept samples up until about 2 p.m. or so, but I would just shoot Gian an email just to let her know. So we have a cooler outside to accept your samples. The samples need to be kept on ice the whole time. So we just want to make sure that cooler is still out waiting for you to put your samples into it. So the Bacterial Action Team, in a nutshell, our overall goal is to protect human and animal health and safety in the water, that's streams or lakes. And one way that we do that is by measuring the extent of bacterial contamination in our waterways. It's when we're talking about human health and safety while we're recreating in water bodies, we're generally talking about bacterial contamination, because, if you get some water in your mouth and you swallow it, that could lead to some gastrointestinal issues, or worse, depending on the kind of bacteria that is in the water. So the first way that we're going to do that is through a visual inspection of the water body for toxic cyanobacteria or harmful algal blooms. So we use the word harmful algal bloom to refer to this, but they're actually not algae, they're cyanobacteria. And so it's a little bit confusing when we're talking about bacterial action team. We are really talking about cyanobacteria. They fit into that goal. It's just that kind of in conversation, people will say harmful algal bloom instead. So to report your visual inspection, you're going to use a website through ArcGIS. It's called Survey 123. You don't actually need to download an app or anything. It's just you type it into your browser, in an internet browser, and we've got in your coolers and your instructions a little QR code. So you can scan that, and that will pop up that form. And we'll run through that form shortly. The next part, of course, is collecting water samples. So you're going to be collecting samples with a few different types of bottles. You'll collect 1 sample for E. coli. That's going to be in a sterile bottle. Of course, we don't want to collect bacterial samples in a bottle that already has bacteria in it, because that's going to contaminate that sample. So we have a sterile bottle for E. coli. We have a separate bottle that we use to measure turbidity, and another bottle that we use for another suite of parameters, nitrate, phosphate, and chloride. So to do that, you're going to collect those samples and transport them to the Watershed Center on ice. So with your sampling kit, if you've already gotten it, you'll see that it's a cooler. And so a very important part of that is being able to put ice into that cooler to keep your samples cold on the way to the Watershed Center. So that's something that you'll want to make sure that you have, is just access to ice every week. And you can reuse that ice week after week if you'd like. The third is measuring water temperature. So this is just kind of a basic parameter that we'll measure almost with every sample that we do. We'll take a temperature measurement, and you will have a thermometer provided in your sampling kit. So it's important to use that thermometer that has been provided rather than, some other, if you have another thermometer hanging around, whatever, you don't want to use that. You want to use the one that is provided because they are calibrated by our state environmental protection agency to make sure that we actually know what the temperatures say. So let's start with that first one, cyanobacterial harmful algal blooms. First, I think it's important to say that cyanobacteria are absolutely critical. They kind of produced life on this planet, and they are an amazing just taxa. They're an amazing type of bacteria. They photosynthesize. Some types of cyanobacteria can actually fix nitrogen from the air, so they can take N2 and convert it into a form of nitrate that they can use to grow, which is a really kind of special thing. However, cyanobacteria are great, but when they occur in huge amounts, when they start to form what we call algal blooms or blooms, that's when things can become a little bit, well, I don't want to say deadly, a little bit more scary. They can start to produce toxins like cylindrospermopsin, anatoxin, or microcystin. These toxins, they are kind of like nerve agents. They can produce a variety of different responses in humans and animals, depending on how much of this toxin is actually in the water. So if it's kind of at a low level and you put your hand in the water, you may, your hand might get a little bit red, you might get a little irritated, maybe a little rash. If you ingest some of it, you could get some gastrointestinal issues, maybe some flu-like symptoms, fever, just feeling kind of like off. At a really high level, if you ingest it, you could experience organ failure, which could lead to death. Now, again, I want to emphasize that only happens when the concentrations are very, very high. So I don't imagine that you are going out to a lake or a stream and just kind of putting a cup in the water and just drinking it when you're thirsty. I would advise against that. But if you have dogs that are going out with you and they get close to the water and they start lapping it up, that's really a more visible impact of the impacts of these toxins. There's been a lot of reports in areas where there's a lot of cyanobacteria, where there are deaths. We have deaths of dogs because people just don't know, or there's no signage up to let you know that there's an active harmful algal bloom. So that's why it's so important. What we're doing here is what you're doing is you are the eyes on the ground. You're looking for these harmful algal blooms. If we do end up finding one, then you are kind of the first link in this chain to be able to report that to the state, to us, to the state. We can go out, collect more data, either confirm that it actually is a harmful algal bloom or not. And if it is confirmed, then we put up signage, then it goes out on a statewide map, and we make people aware, just generally aware, that there's an active harmful algal bloom. So to make a HAB, you need a few things. First, you need nutrients. So you need kind of the food to allow this bacteria to continue to grow. Second, you need sunlight. So they're photosynthesizing bacteria. They need access to sun. They really love really warm water, this kind of hot conditions. And that is also more likely in slow moving water. So HABs are much more common in lakes over rivers. Because you don't necessarily in the middle of the lake have that riparian cover, have the trees kind of covering the center of the stream. It's open to the sun. So you've got the nutrients, you've got the sunlight, you've got the hot water. It's kind of slow. So the bacteria are kind of allowed to just continue to grow and grow and grow. And when they continue to grow like that, that's when they start to bloom and create a harmful algal bloom. So preventing a HAB, thinking about the reverse of this, one big way that we can do this is by reducing and treating our stormwater runoff. So stormwater runoff has a lot of nutrients in it, whether that's from fertilizers or wastewater. There's a lot of anthropogenic sources of nutrients that are getting into water bodies from stormwater. And so that's something that we can control. That's a big part of our whole educational campaign here at the Watershed Institute is teaching people how they can reduce stormwater runoff and treat it so that we can prevent things like this. So I'll go through some pictures so that you can see what HABs generally look like. This picture is fuzzy, but you can see the color of this water. Generally, water in this region is kind of maybe a little tinge of brown. It's not usually like bright blue, bright green. It's kind of just like a natural organic color. When it turns into this green, thick pea soup color, that is a really good indicator that you've got an active harmful algal bloom. So that is something to look out for. We have this on our visual assessment form, and we ask this in a few different ways. There's one question asking about water color. So you can say, is it brown? Is it green? Is it blue? Blah, blah, blah. So that's one indicator to us. Sitting back here, we can look at your data and say, okay, the water's green. Let's dig into this. Another way that we can track this is through turbidity. Turbidity is a measure of how clear the water is or how cloudy the water is. And so if you were to pick up a cup of this water, would you be able to see through it? And oftentimes when we have an active HAB like this, you're not able to see through it because there's so many of these little bacterial cells in that water that the turbidity becomes a lot higher. So pea soup is one thing we're looking for. Another thing are green streaks. Green streaks or paint streaks, we sometimes refer to this. So you can see in this photo, you've got this blue-green swirl on the surface of the water in the center here. So Cyanobacteria are also referred to as blue-green algae. Again, not algae. I don't know why we do this to ourselves, but Cyanobacteria are blue-green algae because they will look blue-green when they are in abundance. And so you can see in this photo, those streaks on the surface of the water are blue-green. A HAB could also produce streaks that are just more green rather than blue-green as well. You might also see a green foam on the surface of the water. That could be a possible HAB, so that's definitely something that you'll want to note. One way that we will capture this is in one of our questions on the visual form, we're asking about surface coating on the water. So that could be bubbles, it could be trash, it could be leaves, or it could be foam. And so you'll want to definitely note that if you see any of those surface coatings. Another possible harmful algal bloom are very small green dots. So it may not be that the water is looks dyed green, but there are bacterial colonies that are growing growing large enough that you can actually see little dots of green in the water. And so that is possibly going to be a harmful algal bloom and something that you'll want to note and photograph for us as well so that we can confirm or not confirm that. Now this is important, very important to note. Talking about harmful algal blooms, we are having a massive one right now. We found this last Thursday, so this has been going on for just over a week now. This is a picture taken on July 22nd by the DNR Canal State Park, and this shows the Millstone River on the left. This is the Beeden Brook on the right coming into the Millstone River. And so at this confluence, you could see the color of the water in the Beeden Brook, normal, natural, regular kind of stream water, meeting with the Millstone River, which is just like a hot green color. And this is just shocking to me for a number of reasons. One of which is that the Millstone River is a moving water body. It's a moving system. So I mentioned before, we usually find harmful algal blooms in lakes or ponds, something that's very still and slow. So the bacteria aren't like, flying around in this system. They're able to kind of just stop and reproduce. In a moving system, that's less likely. However, in the Millstone River right now, we honestly, we don't know what is causing this to happen. We think that it may be growing in Carnegie Lake, which is the center lake in our watershed located in Princeton. It may be that kind of is like where everything's being born, and then it's flushed downstream into the river. But we're seeing this at such a concentration that is just something I've never seen in my life before. So this picture is pretty indicative of that. Here is a picture a little bit further downstream where the Millstone River is meeting with the Raritan River confluence. And so this was also taken on July 22nd, which I think was just last Friday, by the DNR Canal State Park. This was taken from a drone aerial. And this structure in the bottom of this picture here, this is a drinking water intake for New Jersey American Water. I don't know about you, but I don't want to be drinking microcystin toxins. And they don't want people to do that either. So they have shut down this drinking water intake. Right now, they have been talking about alternative options for getting water to their customers. So the Raritan River coming in from the right, upstream of that, there's a reservoir called Round Valley Reservoir. And that is relied on by the New Jersey Water Supply Authority to make sure that we have enough water supply for drinking water. But right now, that water level is very low. So New Jersey is not in a drought right now, technically, but we're approaching drought levels. And they just don't have enough volume in that reservoir to be able to flush this hab out by just kind of releasing the dam and releasing more water from the reservoir through the Raritan River. So we're in a little bit of a pickle right now when it comes to this harmful algal bloom having these lingering impacts on real drinking, on human health and safety through drinking water. This is another reason why this current harmful algal bloom in the Millstone River is just absolutely shocking. So if you have one of your monitoring sites on the Millstone River, first of all, thank you. But secondly, I just want to give you a few extra precautions that you wouldn't necessarily have to take in other water bodies where there's not a confirmed hab. In the Millstone River, you're going to want to wear gloves when you take your temperature, when you are taking your water sample with your bottles. You want to make sure that you're protecting yourself before you put any part of your body into that river. The toxins were measured, I believe, earlier this week, and they were super high. So there's been lots of signage erected along the Millstone River to try to protect people who might be coming here to kayak or canoe or just, have a nice fishing trip. We don't want that to happen right now in the Millstone River. We do want to collect data. So I really appreciate your help in allowing us to do that. But I also just want to make sure that you are being safe when you do it. Another precaution that you might want to take is nowadays we all have KN95 masks or N95 masks. I would encourage you to wear a mask when you go out to sample. There have been some evidence that you can actually inhale some of these toxins, and that's a different way that it can get into your body. So if you wear that mask, wear gloves, and make sure that you take off the gloves without touching, the actual, top of the gloves, then you should be pretty well protected from that harmful algal bloom. So here's some pictures of things that are not a harmful algal bloom, not a hab, but that can be confused for hab often. So one of these is duckweed. Duckweed is a vascular macrophyte. It's a plant that has a leaf. It's a broad-leafed plant. It just happens that it doesn't have a root. So it's kind of floating on the surface of the water. Because it's not rooted, I think people will look at that and say, it's definitely a surface coating of an algal bloom. But if you look a little bit more closely at the duckweed, you'll be able to see actual leaves, like broad leaves in the water. So if you see those leaves, that will be duckweed and not a harmful algal bloom. Another thing that is not a harmful algal bloom are filamentous or stringy algae. So if you're in a stream, sometimes in a lake, if you're able to see the bottom of the water body on this, the benthic substrate, you'll see this filamentous algae kind of clinging to the rocks, clinging to the sediment, whatever's at the bottom, and it's just kind of flowing in the water column like that. If you see that, that is not a harmful algal bloom. That's just regular old algae, but it's important to know anyway. All right, so I'm going to show you what our form looks like, but maybe I should just pause here for a moment. If anyone has any questions, please feel free to unmute or pop something in the chat there. And if not, I will just keep
I had a question, like how would we recognize the kits? Like once we pick up a water cooler, like how would you know that it's from me and then it's from the particular place? Because it can be confused by another person.
Sure, yeah. So you're not, when you come, I'll go over this in a bit, but when you drop off your samples, you're not dropping off the whole kit. You're just dropping off the bottles inside the kit that you filled with the water samples. And those bottles you will label with the site code, that 3 or 4 digit code, as well as the date. So like MR1 7/31/22, that will tell us where it came from and when.
Was that on the email, the code?
It should have been in the email, yeah. When you receive like your latitude, longitude coordinates to access the site, that code should be in the email. But if not, shoot me an email and I can try to clear that up for you.
Okay, and I don't know, the link of the survey that you mentioned in the beginning of your presentation, the one that we're filling out, was that part of the email? Because I don't remember seeing it.
I'm not sure what you're referring to. Link in the survey.
Like once we collect all the once we collect the water samples, we return it. Do we have to fill up anything, any form?
So we have a chain of custody form. Yeah. When you deliver the samples, I'm going to get into all that in a bit. So if you could just be a little patient, we'll definitely get there.
Okay, thank you.
And then we'll talk about exactly what you need to do with those samples when you arrive with the form. So there's some questions in the chat as well. Someone says, do I need to pick up resources before Sunday or would it be fine to pick up the day of? Yeah, so if you haven't picked up your cooler yet, and you could definitely come tomorrow morning, Sunday, as long as you're able to get here, get out to all of your sites and then get back to the Watershed Center by noon. That's totally fine. And then another question, is it okay if we show up earlier to the site than the expected time? Yeah, I think that's fine. I generally put into my timeline 7 to noon because that's like when people are usually awake. But this person said 5 a.m. That is an option. I just want to mention one thing that might maybe complicate that a bit is that when we take our bacteria samples, there is a hold time, which means that when you collect that sample from the water body and put it in your cooler, the clock starts. And we have to make sure that we analyze that sample in our lab within 8 hours of that time. So if you're collecting at 5, that puts us at what? 2 p.m. we have to get the sample in by. I think that's fine. I think that's okay. I would just, if you are collecting it that early, you could write on the bottle itself, in addition to your site code and the date, also write the time, and that will kind of indicate to us when we are looking at 48 samples, which ones we want to pull first to analyze first, if that makes sense. So that should be fine. All righty. So I'm going to show you this form. We can see that. I think yes. Okay. So this form is that QR code on your cooler or in your instruction sheet in your cooler. First, you've got some basic information, your name, your email, the date, and the time that you're visiting your site. Your monitoring site ID, again, this is going to be that code. So it's generally some kind of acronym related to your water body, like Millstone River. Site 1 would be MR1, and iterations of that. When you get to this next part, is this your usual monitoring location? If you're headed to the latitude, longitude, the coordinates that we indicated in the email, then you can just say, yep, this is the usual location and continue on. If you are for some reason not able to get to that site, you're going to click no. And that will bring a map up so that you can pinpoint your new location on this map. And this, for some reason, it's not showing up. Well, anyway, so this map isn't, nope, that's not working. This map isn't loading right now, but it should load for you when you pull it up on your phone. And so what you'll do is you'll just take this blue marker and move it around and place it where you actually are in the field at that moment. And that will just show to us, okay, we're not at our regular site. We're going to enter in these new coordinates to track with the data that you're collecting. So the weather conditions, sunny, partly cloudy, overcast, light rain, heavy rain. This is a multi-select option. So you can select a few different options if the weather changes while you're out in the field. When you're looking at last rainfall, we're looking at least 1/10 of an inch of rainfall. So it's kind of coarse, just say, was it within the last 24 hours, 24 to 48 hours ago, or more than 2 days ago. You can indicate that there. And we'll have, I believe we have a website on the instruction card so that you can refer to that. If you're unsure, like if you just weren't looking for rain in the last 48 hours, there's websites that have historical rainfall totals, and those links will be available to you so that you can kind of check it before you head out in the field, so you already know how to answer this question. For water odor, normal would be just kind of your typical organic stream smell. Anaerobic is kind of that musty, if you step into the sediment and you get that hit of I can't really explain it. It's musty, I guess. You would indicate that. Sulfuric, that would be the rotten egg kind of smell. Sewage, that smells like poop, and then petroleum, that's going to be oil and gas kind of smell. And then other, if you select other, you can write in what that smell is. Watercolor, clear, is just, clear. We want to try to look at the actual watercolor rather than looking at this stream or lake and looking at the benthic substrate. So if the substrate is brown, then that's going to reflect through the water and it's going to appear brown. But when you take your water sample, you'll be able to kind of see in the bottle itself what the color of the water is. So it could be clear in that bottle. It could be green. It could be blue-green, brown, yellow, gray, or white. Or it could be tea stained, which is, when you put a bag of tea into hot water and you just start to see kind of a hint of that brown spreading out, but it's still kind of clear. It's not fully brown and muddy. That would be tea stained. For turbidity, this is, again, the clarity of the water, or how well you can see through that water, or how cloudy that water is. So the more turbid, the more cloudy the water. Turbidity could be clear or no turbidity. Slightly turbid, muddy. So it could be quite muddy, especially you can look at that when you take your water sample, look through that. If you're seeing a lot of particles in that water, then you'll want to indicate that it's either slightly or turbid. That turbidity could look milky. So if there's a lot of fine sediment particles eroded from the surrounding land, you could see that it kind of looks a little bit milky, but also turbid. You'll want to indicate that. And then the green pea soup, of course, if it is turbid and it's due to little green dots in your bottle, you'll definitely want to indicate that. And then other, if just some other kind of condition you're seeing. So surface coating, either none, foam on the surface of the water, scum, which is like kind of algae that's dead and it's just kind of like decaying organic matter on the surface of the water. The paint streaks are what we saw in that photo, just the green kind of swirl. Duckweed or vegetation, leaves would fit into this category. That's a surface coating. You may see oil and oily sheen on the surface. That is here too. And then other. So for water movement, we're using this scale for both lakes and streams. So the descripting words in this scale are going to refer to kind of both. The first term is going to refer to lakes, and then the second term will refer to streams. So lake could be still, it could have ripples, it could have waves, or it could be choppy on the surface, and you can indicate that there. The rivers or streams could be slow, they could have moderate flow, swift flow, or they could have rapids. And so that is how you would define water movement. For aquatic vegetation and for algal bloom presence, you're just saying if there's none visible, if there's a scarce amount, like there's a bit hanging around, but it's not a whole lot, that would be scarce. Moderate is somewhere in between scarce and abundant. And abundant would mean like there's just tons of vegetation or tons of algae in this body. And so, yeah, you'd want to note that the following questions after algal bloom presence will pop up only if you click an option that's not none. So if there's none visible, if you have no algae, then you just hop onto water temperature and did you collect a water sample today? If you click something like moderate amounts, then there's additional questions that will pop up. So if you say that there's algae in the water body, then we ask about what kind of algae it looks like. So that algae could be a biofilm, or just kind of this covering over the rocks and the bottom of your stream or lake. That's one type of algae. It could be that filamentous and stringy kind of algae that's also generally found on the bottom, the surface, or the bottom substrate of your water body. And they could be floating either in globs or scums on the surface of the water, or it could look like that pea soup. That's those floating colonies in the actual water column itself. And then this is a multi-select. So if you do click like pea soup and you do think it's a HAB, you'll want to also click suspected HAB. And that is, that kind of indicates to us, okay, we need to take a closer look at this water body, and then we can head out and collect some additional samples there. For water temperature, please use the thermometer that's provided in your kit. This is reported in degrees Celsius, and that's how it is read on your thermometer as well. So you want to read that thermometer to the closest 0.0 or 0.5. So if it's somewhere in between, like a 0.0 and 0.5 or 0.5 and 0.0, you can just kind of go to the closest value that it is. And then, did you collect a water sample today? This is telling us whether to expect water samples to be brought. And then, of course, remember to keep that chilled on ice on your travels to the Watershed Center. Additional comments, it's just anything else in the field that you didn't answer above and that you'd like to share with us. It could be that there's been a huge rainfall and you're seeing a lot of erosion at that site. There's a lot of sedimentation in that water. Or it could be that, oh man, there's a lot of garbage here that wasn't here, that I didn't see last time. Or there's a lot of people here and there's a lot of dogs and they're pooping and no one's picking up their poop. Any kind of report like that, you would want to comment on that. And then we have photos. So there's a photo of the overall water body. This is you standing back, looking at the water body, taking that picture. This gives us the overall overview of that water body. If you don't click that there's any algae, then that second picture option won't show up. So it just goes overall water body to an additional view, and that's kind of an optional photo. If you do say that there is algae on the stream, then it will come up with a second photo, the close-up of an algal bloom. So if you do find an algal bloom, then just kind of get a little bit closer to the water, take a picture so we can see if there's green dots, if it's the scum, if it's the pea soup, one of those options, then we'll want to see that. So any questions on the form itself?
I'm sorry, would you share the link to go to the form? Because I'm thinking it's on a website, right? I
Yeah,
didn't see it in the email. I went through it very quickly, but I couldn't.
yeah. So the website is located at, do you have your cooler? Did you pick up your sampling kit?
No, not yet.
Okay, yeah. So the link is going to be on the instructions in your sampling kit.
Okay.
So that's what you'll be taking out into the field with you. And you'll only be accessing this form when you're actually out in the field. But I can put a link in the chat here if you want to just kind of check it out ahead of time. Alrighty.
Thank
So
you.
another question, in what situations do we check the suspected HAB box? I really don't want to misidentify the algae. So the suspected HABs are the photos that I was going through before. If the water is dyed green, if there are the paint streaks on the surface of the water, green paint streaks, or if, oh no, I direct messaged that. Or if there's the green foam or globs on the surface of the water. So basically any kind of dyed green situation, you'll want to click suspected HABs. Now, I don't want you to think that if you don't click suspected HABs, that nothing will happen from that. You're still submitting photos of your site. You're still submitting this ancillary information. So if you're saying the water is green and you don't click suspected HABs, we're still going to take a closer look at the photo that you've submitted to kind of see if we can find if the water is green for some other reason, or if it is green from a harmful algal bloom. And same goes for like the surface coating. If there's scum on the surface, or especially if you click paint streaks on the surface, we're gonna think that's a suspected HAB.
Could I have a copy of the slideshow, because I want to look at the pictures again.
Of course. Yep. And so Gian will also send out this recording. So with that, yeah, I'll ask her to send out a copy of the slideshow. Okay. All right. Any other questions on the form itself?
I do. About taking the temperature, how long should you leave the thermometer in the water?
Good question. Really good question. So I would say a best practice would be about a minute to allow it to equilibrate to the actual temperature. Some notes there, you don't want to leave the temperature sitting on the bottom of the stream. You want to leave it like in the middle of the water column. Do I have a thermometer here? Nope, I don't. So there, you want to make sure also that the bottom half of the thermometer, probably bottom third of the thermometer, is fully submerged in the water. So it's on a string, you can kind of dunk it into the water, make sure it's in the water column, not sitting on the bottom, leave it there for about a minute, and then pull it out. And then when you take the reading, you want to make sure, this is a pen, but it's just an example. You want to make sure that you're holding it from the top to make that reading rather than from the bottom. So if you hold it from the bottom, you'll be heating up that thermometer and it will be the temperature is falsely elevated. So hold it from the top after about a minute.
And should we wipe it down? Because if we're taking it to another water site, are we concerned about any cross-contamination in that respect?
Yeah, no, that's a good call. So if you've got water, like just regular like tap water, you could definitely rinse it in between those sites. That would be great. Yeah, thank you. So there's another question, just kind of generally, if I were to pick up the cooler on Sunday, would I need to go get ice afterwards, or will it be in it already? There's no ice in the coolers. So you do have to provide your own ice. The coolers have been sitting out since Wednesday, so that would be a bad time if we put ice in it. But yeah, so you should plan to go get ice after that. All right, I'm going to switch back to the PowerPoints.
One more question. Once we are collecting the sample, sorry, the picture, how big should the picture be? Like of the entire lake, or do I go really close to the water and then collect it? Like take the picture?
Right. So the first picture, this the overall view of the water body. So that one you stand back, and you'll want some of the water body, some of the surrounding land in that photo. So it's kind of just looking back at the whole water body itself. If you click that you've got algae, then that second picture option pops up, and that says close-up of algal bloom. And for that, you'll want to get closer up to take a picture of the algal bloom.
Okay, thank you.
Sure. So after you submit that form, there will be a confirmation page that pops up, and you'll find a link there. If you have a suspected HAB, you can go ahead and report that to DEP yourself. You don't have to do this part. This is basically a very similar form to what you've already filled out, but this is an option. So this is something that we will also do once we go out and collect additional information. If you click that you have a suspected HAB, that if you have paint streaks, if your water is green, we will go to your site within a day or two after you've collected those samples to use our own meter. We have a meter that measures phycocyanin, which is a pigment that's produced kind of like during photosynthesis. And the levels of phycocyanin can be roughly translated to, the number of cells that are in the water. So that's another way that we can kind of confirm if it is or is not a harmful algal bloom. Then we can report your data along with our phycocyanin data to DEP. They come out and they take their own samples, one of which is to measure specifically for the toxins. So they'll look for microcystins. And then if they find microcystins, if they find a very high cell density of bacteria, that's when they start to put up these alert levels. And so if you have a site that's in an area that has a harmful algal bloom, you may see a sign already erected near where people might get into the water that says something like watch or advisory, warning, or danger. So right now the Millstone River is set at warning level. We have toxins that are above 20 micrograms per liter of microcystin. And so the cautions there are that we're recommending against recreation in that water. So primary recreation, this is a term that's used above. Primary recreation is like you getting into the water with your body. That's like swimming or diving or whatever. Secondary recreation is you getting into the water in like a boat or a kayak, something where you're much less likely to actually touch, engage with the water itself. So at a warning level, they recommend against boating. At lower levels, they would say, oh, well, maybe we don't want you swimming, but boating is fine. And so it's just kind of this like balancing of risk versus reward, where they're trying to manage people being healthy when they're out in these water bodies. DEP also has a map of their data and all of the active harmful algal blooms in the state right now. So you can just Google like NJDEP HAB map and it will pop up and you can see the map. There's also a link here. So again, you'll get this presentation. So you can follow that link to look at that map as well. Okay, moving on to the actual sampling kit. We've talked about the digital form. Now we're going to talk about the physical stuff that you've got in your kit. So first, you've got a little blue cooler. This has our old logo on it, but we're using it and that's fine. In our cooler, you'll have a marker. The marker will be used to mark your bottles. Again, you'll want to mark them with the site name and date at the very least, so that we can, when we get all these bottles back at the end of the day, we can know what we're testing. So you've got, I think each of you got 6 of these sterile bottles, and maybe 3 each. This larger bottle here with the green top, that'll be used for the turbidity sample. And these smaller clear tubes, that will be for the nutrient sample. You'll also receive a bag full of gloves so that you can use them. If you have a hab in your water body, you can put them on to collect your samples for all of your samples. But even if you don't have a hab, you'll want to put on the gloves when you're taking your sample for bacteria. So again, your hands have bacteria on them. We want to make sure that we are confining that bacteria and not introducing any new bacteria to our water sample when we are collecting it to measure E. coli. You'll also have a thermometer. It might be purple, it might be blue, but it'll generally look like this. That's an alcohol-based thermometer. It's not mercury or anything. And so this is that bottom part with these perforations at the bottom. This is the part that you want to make sure is submerged in the water when you're taking the temperature. And then when you read it, you want to hold it from the top. So I see that there is a hand raised. If you want to go ahead.
I have a question. Could you go over which containers are for what, and also for the ones with the big caps, how many of those do we need to fill up?
Yep. So I'm going to go over that right now.
Oh, okay.
Okay. So this bottle with the green top, that's the turbidity. The ones with the clear top are going to be for nutrients. And the ones that are kind of bigger and have probably a white label on them, those are going to be for bacteria. So with any of these, you want to make sure that you're labeling it with your marker. Your sterile sample bottles could look like one of these 2 options. Again, they have been autoclaved. They're absolutely sterile inside. And so some of the bottles may have this plastic ring on the top. And that tells us like, okay, this hasn't been opened before. It's definitely still sterile. Other types of bottles that you may have this sticker on the side. And so when you break that sticker, you know that it's been opened and it's no longer sterile. So when you are labeling your sample bottles, you can label it just on the side, on the plastic, on the label itself, anywhere where it's not going to, rub off or anything. Okay, so should we be doing that with Sharpie? Yeah, so you have a marker in your kit, and you'll use that marker to label your bottles. So the first step when you're doing your bacteria sampling, so again, this is the bottle you're using for bacteria sampling. You will want to label that first with the site code and the date, and allow it to dry if the marker is like super wet or anything. You want to make sure that dries before you dunk it into the water. You're going to put your glove on the hand that will remove the bottle cap. So let's see what I've got. I have my water bottle. I'll use that. So if you're taking your sample and you have, your bottle, you have your cap. I am right-handed, so I'll be holding the bottle in my left hand. I will put the glove on my right hand, and the gloved hand is going to be the one that removes the cap. Now, when you remove the cap, you want to make sure that you open it and hold the cap upright. You don't want to touch the inside of the cap at all. You want to hold it from the outside. Again, this is your gloved hand. And then the other hand, you're going to dunk that bottle into the stream to collect your sample. It's about 6 inches below the surface of the water. You don't need to get super exact about that. That's just kind of an estimation. You want to avoid getting into your bottle any big clumps of algae or vegetation or any, actual like living organisms. We want to avoid that. We're trying just to get the water in the bottle. You want to make sure that you fill it up above the 100 milliliter mark. So there's this line here towards the top of the bottle. That's going to be your 100 milliliter mark. We need at least 100 milliliters to run the sample. So you can fill it up all the way to the top, but maybe leave just a little bit of airspace right at the top so that we're able to shake it when we get it. Bacteria tends to clump in the bottle, so we need just a little bit of space to be able to shake it up to get the bacteria equally distributed through the bottle. And then what we'll do in the lab is we will then take off some of that extra water so that we get it to exactly 100 milliliters. So anywhere between the 100 milliliter mark and the top of the bottle. Now, after you have your bottle filled like this, then you're going to take the cap in your hand, in your gloved hand. You have not touched the inside of it, and you just put it right on the top and spin it closed. And make sure that it is tightly closed. You don't want it to open in the cooler and become exposed to like the ice water, or have the lake water be exposed to the ice. And so when you put it into your cooler, you want to make sure it's nestled really nicely into the ice, and not, you don't just want to have like an ice pack in your cooler. That's not going to be enough to cool the sample. You want to get it into the ice and make sure that it really does cool down. Any questions on this process, collecting bacteria samples? Okay. Now for the other samples, these are the bottles that we'll be using for those. For this, again, you want to start by labeling with the site name and date. You're going to use the Sharpie that you have. You could use whatever marker, like if the Sharpie that we gave you, it doesn't work for some reason, we can give you a new Sharpie, or you can just use one that you have at home. That's fine. So you're going to take this bottle. This is different from the sterile sample collection. This is not sterile. In fact, you'll want to make sure that you rinse this bottle pretty well in the stream. So you want to uncap it, dunk it into the stream, pour it out, dunk it into the stream again, pour it out, dunk it into the stream again, pour it out, and then you can take your sample. So you're going to rinse it 3 times. And when you're rinsing, make sure that you rinse the actual bottle as well as the cap. So you're dunking everything 3 times. After you've rinsed it 3 times in the stream water or the lake water, then you're going to do the same thing. You're going to submerge it about 6 inches below the surface and fill it up more than, definitely more than halfway, probably towards the top of the bottle. And then recap it. You know, you don't have to use sterile procedures here. Again, you're just putting the cap on like normal, but you do want to nestle it into the ice so that the sample cools down. And so for each site, you're going to take 1 bacteria sample, 1 large green capped tube, and 1 small clear capped tube. So one of these, and 1 of these, 3 bottles per site. Now, to deliver the samples, you will have your cooler full of ice. You'll have your sample bottles in the cooler of ice. And when you're done with your second, third, whatever your final site is, that's when you will head over to the Watershed Center to deliver your samples. So you want to make sure that all of your sample bottles are labeled and closed tightly, because they're going to be mixed in with a lot of other samples from a lot of other places. You're going to fill out, there's a chain of custody form at the Watershed Center where you drop off the samples. So you want to make sure that you fill out that form, and that tells us when you dropped off the samples, and you are writing your name and signing your name, saying that you've dropped it off, and here are the sites for which I've dropped this off. You're going to wipe off the side of your sterile bottle and use a laser thermometer that will be provided in the cooler to record the sample bottle temperature on the data sheet. So you are trying to cool your samples from your site to the Watershed Center. When you get to the Watershed Center, you're basically going to wipe off the side of your bacteria bottle, shoot the laser thermometer at the side, and see how well you did. And our goal is to get that temperature around 4 degrees Celsius, but definitely below 10 degrees Celsius. If you have a site that's closer to the watershed center than farther, if you're farther away, it will have more of a chance to cool down. And so your sample will likely be colder if you're coming from farther away. If you're right around the corner and you're just driving like 2 minutes from your site to the watershed center, your sample probably doesn't have a whole lot of time to get below 10 degrees Celsius, and that's okay. But you'll still just want to record that temperature and put it into the cooler of ice that's provided. So you have your own cooler, your blue cooler with your samples. You come to that side cabinet that I described in the email. That's the side delivery entrance to the watershed center. There's a cabinet where if you have already found your cooler, you know exactly where it is. If you haven't yet, follow the directions I've given you in the email I sent a few days ago. That will direct you to the bacteria supply cooler. It's in cabinet. It's in that cabinet that you'll find the chain of custody binder, the laser thermometer. So you write down your sites, you'll take your temperatures, and there will be a cooler of ice on the ground. It's a big green cooler. And so you take your bottles from your blue cooler, put them into the green cooler, and then you will leave with your blue cooler. So that stays with you until the end of the sampling season. All right. And then you're going to pick up your sample bottles for the following week. There's 2 types of sample bottles that you'll pick up, or 3 types of sample bottles that you'll pick up. And there's a question in the chat, what is the 3rd sample? Okay, we have a bacteria sample, that's 1 bottle. We have a turbidity sample, that's the green topped bottle. We have the nutrient sample, that's the clear topped bottle. So there's a white capped bottle, a green topped bottle, and a clear capped bottle. Those are the 3 kinds of bottles. You want to make sure that you have 1 of each kind of bottle for all of your sites. If you have 2 sites that you're monitoring, you will want to make sure before you leave the cabinet, after you drop off your samples, you want to make sure that you have at least 2 bacteria bottles, at least 2 turbidity green capped bottles, and at least 2 nutrient clear capped bottles, so that you can collect those samples for the following week and deliver those. Kind of repeat this cycle. So you drop off your samples, you take your sample bottles for the next week. Does that make sense?
Could I ask my question?
Sure.
So first of all, we're getting the same lake water for all 3 of the bottles, right? We're not doing anything different in the actual collecting.
Correct. It's the same water.
Second thing, we're getting our location and everything in our email.
Yes, you should have received an email from Gian with your locations, your site codes, and the latitude, longitude.
I don't think I've gotten that.
Okay. Sorry, who is speaking? I just want to make sure.
Brandon.
Okay, got you. All right. I will.
My parents' email. I don't have it.
You don't have my email?
No. I think I
Oh,
got it.
okay, got you. Okay, you need.
I found it. I didn't look hard enough. Sorry.
Oh, no worries, no worries. Okay.
I had one more question. I put my question in the chat window. So will you be sending this presentation to us?
I'm so sorry. I can't hear you. Really fuzzy.
Oh, can you hear it better now? Can you hear me?
Barely. Can you try again?
Yeah, I put my question in the chat window. If you will be sending the presentation to us.
I don't know. If someone else knows, I don't know what's. I'm so sorry. I can't hear you.
You will be sending the presentation to us. That was her question.
Yes. Yeah. Thank you. Thank you so much. All right. So let me look back through the questions here. Okay, so we have a question. Is it possible to watch the process of analyzing stream samples at the watershed? For that, Gian will be doing the lab work, so you can just shoot her an email and ask if you can come in to check it out. I think that would be fine. Okay, so do we complete the same rinsing process for both the nutrient and turbidity samples? So yes, for the green capped bottle and the clear capped bottle, let's go back here. For both of these kinds of bottles, this top one is turbidity, that bottom one is nutrient. You're going to triple rinse these bottles in your stream and lake water before you fill them. It's for the sterile bacteria bottle that you do not want to rinse it. And for that, you want to make sure that you're using sterile procedures. You're not touching the inside of the cap or anything like that. How to obtain the nutrient sample with the clear top. So for both of these samples, oh, sorry, yes, got you. All right, so for both of these, yes, you're triple rinsing and filling it up. Sorry, can you repeat what we need to do to rinse the bottle? So you uncap it and you put it into the lake or stream water and you fill it and then you dump it and then you fill it and dump it and then you fill it and you dump it. So you're just rinsing the bottle with the lake and or stream water before you fill it up with that water. Gotcha. Okay, cool. And okay, another question. Do we go to both sites or do we pick one? Yeah, you're going to go to both sites or 3 sites if you have 3 sites assigned to you. Those 3 sites you're going to visit week after week. So it might be a good idea to just kind of plan a route from your house to each of those sites to the Watershed Center. And you'll continue to return to those same sites for all 5 weeks. Right. So when you get to, so 1 site per week, no, you're going to go to all 3 sites, all 2 sites every week. All right, cool. So the chain of custody form, when you get to the Watershed Center, it's in that cabinet that I described in the email. This is the chain of custody form. It's going to be in a binder. And when you arrive with your samples, you're going to start by writing down your site ID, which could be CL2 or some version of that. The number of bottles, again, you're going to have 3 bottles per site, your bacteria bottle or your sterile bottle, your turbidity bottle, and your nutrient bottle. So when you take the temperature of your bottle, you're going to wipe off the side of the sterile bottle or the bacteria bottle. That's the same bottle. You're going to wipe off the side. There's a towel in the cabinet and you're going to measure the side of that bottle. So you're not like opening the cap and sticking a thermometer in it. That would contaminate the sample. You're going to use the temperature gun to point at the side to collect that temperature. Then you put the time that you're delivering the samples to the Watershed Center. So, look at your watch, whatever the current time is that you're dropping it off, and then do your signature. And then you'll repeat that for your second site and third site as you need to. So this is what the inside of that cabinet looks like. We've got some extra supplies with bottleware, extra nutrient bottles, which are the clear top bottles, extra turbidity bottles, which are the green topped bottles, and extra bacteria bottles, which are the white topped bottles. We also have, we'll have extra gloves in case you are going through them more quickly and you just need to replenish those. Those will be available there. So here's a look just at, inside the tray where we have the bottles that you'll pick up for the following week. Sometimes the caps will be on, sometimes the caps will be off. You just want to make sure, of course, that you have a cap that fits your bottle appropriately. And then here is a video of the thermometer gun. You really just, there's a laser and you will basically pull that trigger and point that laser at the side of your bottle. When you take your finger off of the trigger, that temperature will hold. So you don't need to continue holding the trigger to read the temperature. You point it at the side of the bottle, take it off, and whatever that temperature is, however that reads, that's what you'll write down on the chain of custody form. All right. So the volunteer expectations for the bacteria program are first and foremost to keep yourself safe. you're going out into the fields, there could be situations where we have very heavy rainstorms and it's really muddy and slippery on the bank and it's hard to get into the water. In those cases, I would like you to prioritize your own health and safety over grabbing a sample for us. You know, we just want to make sure that you are being careful while you're out there, and collecting a water sample like in a hurricane or something, that's not worth it, you know, so don't do that. And just make sure that you're staying safe and healthy. Second, please take care of the equipment, take care of that kit that we've given you, and please be sure to return it at the end of the season. So there's 5 weeks, 5 Sundays of samples. On that 5th Sunday, rather than leave your bottles in the cooler that's been provided, you're just going to leave your whole cooler. And so we'll just make sure that we get everything back from you at the end of the season. Our goal is to get you to monitor every Sunday of the 5-week session. There is a state standard for E. coli measurements that we get 5 samples within a 30-day period. And from that, we take the geometric mean of those results, and that provides us with the score for how you know well that stream or lake is doing in regards to its bacterial contamination. So we need the full 5 weeks. And then you want to make sure that you get your samples to the Watershed Center by noon, collect samples, you know, we talked about that. You can collect them a little bit earlier if you'd like. And then be sure that you're following the directions for the sterile sample collection, making sure that you're not touching the inside of the cap or the inside of the bottle. You're wearing gloves on especially the hand where you're taking the cap off. And for the turbidity and nutrient samples, the green topped and the clear topped bottles, that you're triple rinsing those and making sure that they're nice and rinsed before you actually collect the sample. So here's your weekly routine. First, what I would suggest is checking the local rainfall totals before you leave the house. You want to look at the last 24 and the last 48 hours. So that will help you to answer that question in the field. If you don't know if it's rained, you can just check that website and that will be able to tell you. If you know, you don't have to check the website. You can just say, yeah, it rained. I saw it. You want to make sure that you fill your cooler with ice before you leave the house. I suggest that you label your bottles before you leave the house as well. It's just kind of easier to jot down the site names and the date on all the bottles using your marker. And then you can just pop them in and go. And all you need to do in the field is just dunk them in the water. And that should be good. You want to make sure you hit all of your monitoring sites on your itinerary each week by around 11:30, so that you're able to get to the Watershed Center by noon to drop everything off. At each site, again, you're going to arrive. You're going to do your visual assessment using the QR code or the link that is provided in your sampling kit. You're going to measure the water temperature using the thermometer that's in your sampling kit. And you're going to collect 3 water samples per site: bacteria, turbidity, and nutrients. You're going to put those on ice and transport those water samples to the Watershed Center by noon. And then when you arrive, you're going to put your samples into the cooler that's provided, complete that chain of custody form so we know who's brought their bottles and what the temperature is of that bacteria bottle. That's pretty important. If the temperature is above the threshold that we're looking for, that could have some impacts on the actual data that we're getting. We might see some falsely elevated bacteria levels if it's not cooled down enough. So that's why it's important to record that information. Alrighty. So that is that for me. Are there any final questions before we log off here? And again, you will receive a copy of this recording and you will receive the PowerPoint as well. So you can refer back to this if you need to. And if you haven't yet, you can come pick up your sampling kit today. You can come get it tomorrow morning. Just make sure you have it before you head out to your sites. definitely.
Thank you.
Thank you. And so, yeah, if anyone does not have their site information, so with the actual sites, you'll need a few things. You'll need the latitude and longitude, so you know where you're going. You'll need the site codes. So, you know, those codes I was talking about, MR1, CL2, VD4. If you don't have those two pieces of information, can you please email me and I can make sure that you get the information that you need. So you're set up for success tomorrow. And you can just reply to my email. Okay, there's a comment, and you said to pick up sample bottles each week, but we need 3 bottles, right? What about the bacteria sample bottle? Yep, so each week you're going to need to pick up the bacteria bottle, the turbidity bottle, and the nutrient bottle. So you have your first week enough bottleware, I believe, to do the first 2 weeks. For your bacteria bottle, I think you might need to come in and with your first week, pick up your turbidity and your extra nutrient bottles for the following week, but you may have enough bacteria bottles to get you through the first 2 weeks, you know? So you just kind of have to be conscious of like how many bottles you have in relation to how many sites you have so that you've got enough stuff to do your sampling for the following week. And like, you know, if you forget to pick up your bottleware when you're dropping off your samples, I mean, it will still be in the cabinet, and the cabinet is open. It's external to the building, so you can always come by later in the week and just say, ah, I forgot my turbidity bottle. Let me stop by and pick it up. If you're living far away, you know, maybe you don't want to do that though. So you want to just make sure you grab everything that you need. Okay. If you, go ahead, Kathleen. Yeah.
Sorry, I just wanted to bring up the thing about marking our volunteer hours.
Yes.
That.
Yes. So for hours, I believe you'll go to the watershed.org/hours. And that will bring up a form that you can submit your name, the activity that you're doing, which is StreamWatch, and then the number of hours that you're working in the field. And so when you're including your number of hours, you want to include by the time you leave your house, all the way through collecting all your samples, delivering to the watershed by the time you get back to your house. So it's basically door to door to and from your house are the number of hours. And then I'll make sure that we send that link out as well so that you've got it. All right. Yeah, a note in the chat. Thanks for mentioning that. The cabinet door is usually jammed. Yep. So it opens from the left side. You just got to really yank it. It's never going to be locked. You just gotta pull it pretty hard. So thank you all. Thank you so much. This is a huge thing that you guys are doing for us, and we really, really appreciate it. And so if you have any questions along the way, if there's something that's kind of like in the back of your brain later tonight, please feel free to email me, and we'll try to get all of your concerns addressed by tomorrow so that you've got your stuff, you've got the idea of what you're doing, and that tomorrow we'll just have a nice sample day. Hopefully the weather is nice as well. All right. Well, I will stop there, and I hope you all have a really nice rest of your day. Thank you so much.